Suspended. Yes, so we are going to centrifuge. Are you done with suspending? Like I said, we've optimized some protocol, so we need to just go at the same pace. Is that okay? So right now, you have to start that again. You've already repeated. What happens? So the faster we are, the better, so we can move on to the next stage. Because I try like those things and so we're going to resuspend, we're going to centrifuge the resuspended microbe in the water, liquid free water. We're going to resuspend we're resuspended already, so we're going to use the centrifuge to separate the pellets from the supernatant. So that's the next thing we're going to do. So we'll make use of the pellets for extraction. So have we already suspended in the tube? Have you done yours too? Yes. Are we ready? So we just have one person representing protein. Then we'll want to centrifuge and come back to protein. So let's have all your tubes in one rack. So we're going to centrifuge the resuspended cells in the water. We're going to recentrifuge to have the pellets. So it's the pellets we're going to be making use of for extraction. Where's the fifth tube?
No, it's there.
So we're going to be centrifuging
12 grams. 12.
So we're going to be centrifuging at 16,000 RCF, for 5 minutes. suspend itself when you continue like that. So we notice that pelletizing makes it easier for us. Yes. Exactly. To be more concentrated when you use the pellets. Okay. We will. What? So what's going to happen now is we'll just move to. So we'll just. So let's go. You can take some from here. Okay. Come and open it. Like I said, we are trying to resuspend itself. This place is called. This one. You have to discard India.
How do you get the tailor? Is it not the one that you don't start is you can actually discard the paper. Adding from them a picture of from the picture of the biological reaction method. Because adding everything inside the yes.
So now wait for 10 minutes.
Yeah, it's basic for 10 minutes. So is there any order I can add it on the order you produce it first or you can add if you are using this one, then you are using yellow. If you are using this one, so that we won't contaminate the whole service. The whole kit. That's why we are so what?
Oh, it's already giving us.
The one what you're supposed to provide one for this table, one for that table, so that we have the date for control. So that we can
I'm counting it now.
It's not inside the
yes.
You have your house here. You check it. You allow it to. Now you get it. And it's there now. You have your house here.
So that one is 2
So can you help me delete it? Don't know what.
I've already told you about I've already told you about
Like, I will just pass everything. Don't worry, I'll pass it on to you. Yes. Before integration, you adding to the indicator. 20, I think that. I'll pass it on. Let's pick this one No, I was thinking it's 6 is also
So I added to this.
yes. After we get this, after we get this, we are integrating. As we start with this,
Let's do it together.
yeah. My God, let's show that.
The first one, 20.
Just 200 of this. Have you had that? So you have to
Yes.
we go before tax, we now need settle. So just bring everything down. Let us know.
It's not one first.
So you incubate for how long? Let's check your manual here.
You set, you already set.
So this is time. Yeah, so I'll set a timer for it. So some of these things, while you do your extraction, it's nice to always set them up even before you start. I had to set this up for the cake so that you imagine that the temperature has to rise to 55 degrees. It means that it might not get there even before you get to the stage. So before you start any extraction procedures, it's nice to set up everything you want to use, from your pipettes to your tubes to everything. Ensure that you have everything in place so that you don't have to move from one spot to another to ensure that your extraction goes smoothly.
Right. You're right.
So like I said, we'll go straight to PCR after this. We'll don't have our PCR session after this. We'll prepare the setup. We'll continue with the theoretical part of gel electrophoresis and come back to the lab to have that. We'll see how far we can go to tomorrow. We'll continue.
Okay, so you said for 10 minutes.
10 minutes.
So where is the timer?
This is the
This
time.
is the timer. So this is here.
Still have 8 minutes.
So once the time is up, it will be. Yes. So I'm sure these things are getting clearer.
It's a standalone thing.
Yes,
Yes,
after this, we
yes.
now have
You people sell it like
1 molar
No, we don't sell this.
genomic pineapple.
it comes out.
Well, we can have it.
Genomic pineapple.
Because it's plenty.
Yeah, sure. So the person who won the pipette, so this is a kit. These are all kits. Like I said before, you have different kits specific for different kinds of samples. If you notice, you have this is the kit we are using currently. This is a universal kit. It works for a wide range of samples. So if you are working with specific kinds of kits, if you see this also, this is quick DNA viral kits. So depending on your type of sample or your sample source, specific specific DNA extraction kits will determine the sample will determine the specific DNA
But
extraction.
there was one you were talking about yesterday, about the one specifically for environmental samples.
Yeah, they are kits for environmental
Yeah.
samples.
So
That's
which
the
one is
Zymo
that?
Biomix kit.
Okay, biomedical.
Yes, Zymo Biomix.
And it's different from the normal DNA fungal bacteria.
Yeah, sure. That's definitely different from it.
I think we have a small one.
Yes, so this is it. This is the Zymo Biomix kit. So you use it for environmental samples. Soil, for guts, and water and all. So this is strictly for environmental
And
samples.
all of them are 50 preps.
Yeah, they come mostly in 50 grams. So this
But
is,
these small ones, what are they?
there are different types of kits.
I mean for environmental samples.
What?
For environmental samples.
Yes. These kits, this is fungal bacterial kits. So it works for bacteria and fungi.
So,
So the extraction kit we are using can work for blood samples.
okay, this one irrespective of where it's coming from.
Yeah. As long as it's fungi or bacteria, you're trying to extract DNA
It
from.
doesn't matter whether it's from
Yes.
environment or from
No,
chemicals.
if it's environmental, it is advisable to use the kit that's specific for environmental samples because they contain a lot of inhibitory compounds. So it's best to use this. But if you're talking of bacteria or fungi, this already covers. So there's plant seed kits you extract from plants or
No,
seeds.
the question I'm asking is, like this one, it may be fungal, it may be bacterial, but it's from the environment. That's the question. It may be from the clinical setting. So that's what I'm
It may be
saying.
from the clinical
Yes,
setting,
yes,
you can
yes.
still use these kits.
We also make for the particular
As long as it is the organism you're
Organism.
extracting from. If it is the sample you're extracting directly from.
So I've been using this all along, but I would like to know.
Bacteria.
Yes, so
Okay.
since you're telling me about this, so I'm now.
So you want to extract a community of microbes.
Oh.
Metagenomics.
Yeah, metagenomics.
Okay,
That's why you use this kit, exactly.
Oh, I see.
Yes.
But if it's not that,
If it's not metagenomics, I'm just bacteria form.
so I can use this one.
No, it's different.
You can't. Do it different.
No, it means this one is more, this one is more affordable.
This will be more expensive because it's for environmental community.
So which protocol are we using right now? Is it
We're
from this
using,
or-
no, it's not from any of these, it's from this. Remember I said we're using a universal kit. Yeah, so we're using this particular kit, D46 It's a universal kit. You can extract from body fluids, you can extract from microbes, you can extract from tissues. You can extract this is 50 preparations. So most times they come as 50 preparations.
Is it affordable? Because in Nigeria we're looking for something that is affordable. Is it affordable?
It is affordable because it's for 50 people. Okay?
No, but other ones are 50 grams.
Yes, they are all affordable.
Then
And we will have to give you proof.
around 200. Usually that's what we use.
We're looking for a strong one that can see the whole thing. That's what I'm found.
So the thing is, how many samples?
For viruses, we make use of Jenna.
Let's say
It's very
50 samples. So it's actually best to go for this. Why do you want to go for? The thing is because you and so you can go for this instead of this.
But the
If
problem
you
I
are
have
working
is
with
this.
only bacteria, is this.
Okay, this is just for bacteria.
You can use it for bacteria, mostly for bacteria. You know it's a universal kit. You can use it for a wide variety of samples. Okay. But the issue with this is, of course, because this is more specific for microbes, it's safer to use this for microbes.
So including viruses.
Fungi and bacteria.
The small one.
No,
The universal kit.
go and use the kit that is specific
For coronavirus.
Because I've not used it for viruses.
Use it use the viral kit for viruses. Use the bacterial kit for bacteria.
The problem with this now is maybe this is low.
Okay.
Yeah. It's not everybody that has it. So when you don't have one
Device.
then you want
So
to
now, what we use water baths in our lab, so both is not the same for
You can compare the results and find out if they're different.
when you say improvise, what other options do you have?
Another way to keep them. What of that is an example?
Yes, apart from that.
So once we're able to stabilize the temperature like 55 degrees.
Yes, once you're able to stabilize the temperature. Yeah. No, the water bath, the difference, water bath. So it's a bath, it's like a bowl kind of with water inside. So the water is heated up to the desired temperature. And you place your tubes inside. No, this is a dry block heater. A dry block heater.
Is it also incubator?
No.
Incubate just
You're leaving it at a particular temperature. You're incubating because you're leaving it at a particular temperature. You're not doing anything, so it's just staying at a particular temperature.
This is actually not an incubator.
It's not an incubator.
No, she was thinking.
No, it's not an incubator. It's a dry block heater.
This is small.
Everything is just clean up.
That's why we have
This is small. Just millions of dollars in our lab. First, set up the whole piece.
step by step.
So once the 10 minutes is up, We will proceed. So what's your next step? What's your next step?
So we had one volume.
So one volume, so sometimes you see one volume, you see 2 is to 1. So the one volume says if you had, for instance, 400 microliters in your tube, you should add 400 microliters of that the genomic binding buffer, one volume. So if you, it's just like saying add the same volume of what you have in your tube. So if you have 200, you add 200. If you have 420, you add 420. So it is time you take out your tubes. So what's your next step?
We have one volume.
One volume. So you're going to add 200 plus 200 plus 20, that's 420. So you're going to add 420 microliters of genomic binding buffer. Wendy, let me know. So take.
That's the genomic.
They're taking one. They're taking one.
I dropped it here. Madam Edith, you have to add genomic binding buffer. So when you're using your tape holder, ensure that it doesn't touch anything, your gloves, your surface, cause it to contaminate it. So now that, don't use it. Take another tape. This has a tape, take another, eject it, take another.
What do you mean by one volume?
One volume says the same volume as what you have in your tube. So if you have 450 in the tube, or everything you had added together is 450, you should add 450 of the next reagent or the next buffer, which is the genomic binding buffer. So but you have 420, 200, 220 as 420, so you're adding 420 to your tube.
Please, can you pass the This is very
It's not easy. Put yours. Okay. What else?
transfer. It says transfer mixture. So
Let me
just
go to
so just wash it. This one. This one take it. Contract.
So you're going to add it to how many is that? Once it touches the surface or touches anything, it's moist.
That's 105.
That's an example.
Okay.
So it says transfer the mixture to a speed IICXMR column in a collection tube.
Can I have
Remember, I
another?
already showed you. Ensure that your tubes are labeled, please. Yes, so please. Where's your? I get everybody. Is this yours?
No.
Where's yours? Alright. Label it. So what's your next line of action? You are transferring mixture. Hold on, before you proceed. So your total mixture now, your mixture, the total volume is 840, right? 420 plus 420 is 840. The maximum capacity of the spin column is 800 microliters. It means that you cannot add everything at once. Are we listening? So you're going to add 800 first and come back to add what is left. Is it clear? The maximum capacity of the spin column is 800 microliters. It means that you can't have more than 800. Remember, the total volume of what you mix together with that mixture is 840. If you are going to add everything, a bit of it will spill. So it's nice to just have 800 microliters. So I want you to add 800 microliters to the spin column.
That's this one.
Yes, the spin column in the collection tube. Add 800 microliters. So leave it, let me see if it's not too much, because of course pipetting error is good. So it's fine. As long as it doesn't, and it's fine. It's okay, please. It's okay. You add the rest later. If you make it too cold, to spill. Thank you. Hold on. Are we good? Have you added? Have you added? So who else hasn't added? Have you? So ensure that your spin column is properly capped so that upon centrifugation it doesn't spill. Alright? Let's put it in the centrifuge.
No, inside this.
Yes, inside the spin column, but it has to be. Yes. So it has to be something from this. Yes. Change your tip, please. Change your tip. And always ensure that. And anytime you're not using your tips, please cover. Cause it contaminates. So if you add that, ensure that your tube is properly capped. Are you? Yeah, let's go. Let's go to the percent. So what's the speed you're using?
12,000.
12,000 was
For
RCF.
1 minute.
1 minute. So what's the full meaning of RCF? There's one more person I'm waiting for. What's the full meaning of RCF? RCF.
ZRCF or RPM?
RCF. RPM is RCF, but we are using RCF. The G you have there is RCF. How many minutes?
1 minute.
I already mentioned why this has to. It has to get that So the next step says discard the flow through. It's also nice to relate on what the next step is, so you don't have to back and forth.
But I still have this thing on this one. How come?
Yeah, so you add it again and cut it. So whatever you have left, you add. It only makes things easier for Okay. What is this? MSA, MAG, IN, B. Yes. Is this yours? All right. So if you have more, please add it to this finger.
Can I discard the
Yes, you can discard the flow through in the conical flask you have in front of you. The flow through is the liquid at the bottom. This is the flow through. Discard in this tube. If you have more, just take it out and add it. If you've added yours, just bring it. Okay. This is have you added yours? Who else? So we'll do the 12,000 again for 1 minute. The next step says add 400 microliters of DNA pre-wash buffer to the spin column in the new column. So what's going to happen after this is when you discard, you change the collection tube to remove the spin column and place it in the collection tube we provided. Remember we gave you 2 collection tubes. So you just transfer it into the collection tube. Okay. Are you done? Pardon? Okay. And the SA. If yours is here, business romantic. The next step, you're adding. Yes, just remove it. Remove your spin column and place it in the new one. No need, no, just place the spin column in the new collection too. You can leave that aside. Set it at 400. That's what let us can use it.
Can't set it as 400 as well.
I have 400, I'll send it.
It's here, so just hold on a bit. So DNA pre-wash for. Yes, that's it. Just be careful. Go again. Dispense. Let your teeth. So straighten your hair. Yes. Straighten the pipette. So next time, go reasonably a reasonable depth into the tube before you take it. If it's too full, you do just ensure that you go a reasonable depth so that the liquid does not go out before you are done aspirating.
No, don't worry, you don't need to pick it. Don't look at it. When you want to pick it,
Yes. Have you taken yours? Let me have it. Not for the same time.
This is very stressful.
So you're centrifuging again at 12,000 for 1 minute. This is actually a very fast procedure.
Yeah, that's what I was telling you. Like that's in my lab. We don't use kits for tuberculosis because of the fact that it's very resistant and we try different things. So the only thing that eventually I do work out was the sit-out. It's very rigorous. So we spent 2 days.
you have to
Because
take
you
care.
date, you refrigerate for an hour, you take overnight again.
Don't use this.
Who else is left?
Use it.
400, yes.
That's it.
Ensure that you have everything in your hand before you take your tea, cause of contamination. very important.
So, Mr. Uchenna, when you were doing presentation, you said that you could also design primers, like you could use primers.
Just synthesize primers with bullets.
Oh, so what's the difference between?
So designing, synthesizing means you're trying to make. Okay, you give us a sequence and we make it.
Okay, but let's say I want a particular primer for a particular
Organization, yeah, for a specific target. Okay, well, once you give us the sequence, so designing means you maybe see a sequence and you want to design a primer for that particular specific target. So designing primers because you know your objective, you know the target. So it's wiser for you to design yourself and give us to synthesize. Yes. So if you for some reason you have to find it. But when you also design a primer, you can blast it. Primer blast.
So designing a primer is just like it's wet lab, it's just
No, it's computational. It's computational. Dry lab, yes.
but synthesizing is not the wet lab.
Yes. So the primers we synthesize comes lyophilized as freeze-dried. So you have to resuspend the buffer or so primers that resuspend the buffer and last Yes.
Than the one you leave like that in the like
No, the one you resuspend with water.
So that's going to be your stock.
Yes, so you can dilute oil. Your working solution.
And
You always
it's part
put
of
working
it's
solution
part of what
from
you're
C1,
covering.
B1 goes to C2.
It's
Yes.
part of what you're covering for
What?
how to resuspend. What? That's the key now. But you will not be able to yeah, but it's still explain. If not, there's no point. There's no point. You understand what I mean? If I have you're a really bold man. She doesn't know what she's saying. You're having pizza and you cannot handle your time. What are you doing?
So the thing is this, if you must reconstitute, for every synthesized primer, we will always give you a synthesis report. All you need to do is follow strictly your synthesis report and you not have to.
It's just like
It will always tell you add so amount, so volume of
The volume
buffer,
for reverse and for the forward. they are
exactly. So you will always place it in the synthesis
straightforward.
report.
It's really straightforward.
Yes, it's not something difficult at all.
Yes.
So it is
So you're adding what's next? What are you adding next?
700 Napolitan of you know what buffer. So we're
that's
working
the
for
number
715.
number 6
The first one is 7, the second one
of
is
GDN wash.
Okay, that one would be
So just discard the flow through. Discard the flow through.
genomic was fine.
yeah. Is it this? No, wash. Grinding. Wash buffer.
Genomic wash buffer. Remember you've used the
Grinding buffer.
Binding buffer.
so remember to discard your flow through before you add your genomic DNA wash buffer. That's what you're doing. Genomic DNA wash buffer. So we're doing that step twice. 700 microliters, yes. Are you done? Yeah. Wait.
Sorry, can you?
Straighten your hand. You don't bend your chin. I understand that. Let me see what you're doing.
Use this one, use this one.
There's another one here, sir. What's your next step?
The 700 microliter.
All right, let's do that. If you've added, let me have it.
Not easy.
Yeah, I still work with samples.
You see that? Me, I don't know if you give that this
I do it standing. Last Saturday, I stood up for 2 hours. I was teaching and then also doing.
Okay, yes, you had your training last week, like 2 weeks ago.
We had training last month.
Okay, last month.
Last month. I'm not going to be doing next week. So
Next week.
Friday, Saturday. On the graduates.
Supposed to drop something. Please remind me, I need to confirm this. So while this is spinning, what's your next step?
So we are going to have 200 microliter
Of the same. How many?
One minute.
12,000.
Yes. Usually it's the same speed you use all through. I was asking what RC is. Somebody has practiced. Yes. Let me say something.
Do you
have a
I only
response?
know RPA.
So what's RPM?
Rotation per minute.
Okay. Yes, rotation per minute. Yes, what's the alright, I'm not the only. I will ask you that.
But I don't think RC, G, and RPM are the same.
No, they're different. RPM and G, which is RCF, they're different.
So there was a particular time when I first started doing, there was a particular formula that you need to
Yes, yes. The G is actually that RCF. You hardly see it as RC, you see it as G. So it's RC. G stands for what? In physics.
Gravity.
So now use that word.
I'm trying to remember there's this.
Reality centrifugal force.
Yeah, I'm saying the second.
Okay, I know it.
Maybe I thought it's
It's because of that. Well, sometimes.
It's not easy. Tell us I'm just seeing the circle, the centrifugal and centrifugal. So I'm seeing it.
Everything has been provided. Anyone that's not provided, you will see not provided in brackets. To make up quiz and all is not provided. Yes, you have to add it. You can add it to make up quiz. have to beat
Native centrifuge.
I think it's because
Sound the functions has
relative centrifugal force.
Force.
If you
That
had
is
it,
gravity.
if you had it, and you
Yes.
don't have the
Yes.
technology.
So microbial feels like a highly concentrated antibiotic. Yes. Your next step, you're adding 200 microliters. The faster we add, the better.
Yes, the start.
Yes, discard the fluid here and add your 200. How have you guys been using that pen? It's not good. The blue one. The 1000 microliter. No, it doesn't work in the USB. So what do you guys doing? Like when I touch it, interesting. Okay. So we'll see. We shall see.
After this one,
After that, what are you doing next? Are you sure?
after centrifuge.
Yes.
We now have,
DNA wash buffer for 200.
yes, then we centrifuge.
Oh, whenever you centrifuge, we wash first on that.
No, after we use the centrifuge now, after we now have
Yes.
the addition buffer, we are going to increase it for 5 minutes.
We cannot rest with it.
Yes, can go.
Yes,
It's
sure.
so expensive.
That's a lot. If you've added, let me have your tubes. Thank you. Who else? Have you added yours? Have you added yours? No.
We use this now.
Use this.
There's no one there. There's a
No, it's better to use the yellow one.
I've already seen
Once you are very set.
You remember that our teacher? Yeah. That was what he taught us.
Before you do anything, ensure that you are done before you take your tea. So that you not touch any surface. To avoid contamination. You know, once it is contaminated, it's very likely that it will not be fit for use again.
For pressing. The pressing, yeah, the basic.
Perfect. So is there anything different than what you guys do?
That's what it is.
But tell me, your experience, what has it been there? If you had to confess in your candid opinion.
Okay, the experience here is because I have been to work with actually different centrifuge machines.
Yeah.
Over there we use water baths, now we use
Dry blood heater.
so even the water sensor we use over the different water. So and even the kind of microbials I use over the equipment we have this.
Yes, sir.
And the explanation you'll be making even gain a lot of stuff.
Thank you. 12,000, 1 minute?
Yes.
Alright. So there's another step we used to introduce before emission. It's also something we optimize. It's called the dry spin. So dry spin is we spin an empty spin column without any solvents or any reagents at all. We spin it for I think 5 minutes. Yes, just to ensure that you get rid of residual solvents. That's just the function. So you get rid of anything that is left in the spin column, any liquid that's just in the spin column. That's the dry spin method. It is not part of your yes, so but it is something we've optimized over time, and that's why we use it. Okay, so after this step, before we do the elution, we are going to do dry spin.
Okay, before the last step.
Yes, before the final
I think
step.
we do that with Zymo Biomix.
Zymo what?
Zymo Biomix. HRC says there's a particular part for further purification to remove any form of PCR inhibitors. So
This one is very nice.
Yes, not speak.
Matter of time and patience.
Or imagine working now, working with your options, like 30 seconds.
you split it.
No,
Place it on this one.
Thank God you will
Sorry.
No. I hope you have been commending it before for all the tips.
I take like 12. So
If
we are working
you
with
can,
samples,
yes.
You have to just split it.
So that takes lots of time. Like our own century can only take I think
So the question I'm asking is,
So just transfer it into your 1.5 ml.
you're recommending the dry
Into your microcentrifuge tube. Just transfer it. Yes,
before the elution?
if you want to. It's not a must, but you still have your equipment. Yes, so transfer it into your empty tube. Yes. Label, please. Label.
Which tube is that?
The empty 1.5 ml tube. Which tube do you have? Let me give you 1.5.
This?
No.
The
Follow.
How do you want to transfer that?
No, you
Put
just
it, you just put it here.
and discard it.
It's confusing. I think I'm not getting it.
No, you transfer this.
Hide it. We're supposed to discard the phone.
Then discard that. You remove this. Put it inside your.
A new one that is bringing. Okay, so by the time we do that, we're now going to dry spin before we elute. Hey, this is beautiful.
It's good when you have all this equipment and there's light.
The challenge now is the big block.
This one. Light, okay, block.
Yeah.
Maybe
You can make use of
You can make use of it.
water bath. Once you're able to stabilize the temperature at 45 degrees, you just time yourself. So before you start anything, you must have
This one. No.
Sorry.
it should be close to
One.
What should be closed?
This one.
the
Dry block inside. Maybe there's one in the institution have the money. And you know what to
But when they have another
do.
option,
Private university or any private institution, they can afford this than federal. No federal, they just do
we have private institutions that have it, but they don't use it.
So, like I said, we're going to do the dry spin now. Please take it.
okay, thank you.
You label your tube appropriately. And we'll go back downstairs after PCR to do the gel electrophoresis. Sorry?
I need for the 1.5.
Okay, I'm supposed to give you.
Not everybody.
There's mark over there.
when I'm open for it, I actually spoke to them.
When?
That was last week.
I wanted to get one.
Yeah. And this is cool. I just started making my phone. It's just like so used to it. How about taking this class? Maybe try to have some checks.
Cannot just change. It's
Because
very
it didn't
good.
literally work.
It's very good
It was
to
amazing. I don't know if it's
something.
very
No, it's very humble.
It's very long. No, I like if I approach it, it doesn't mean that I approach
You
it.
can get
It's a very simple thing.
where not saying he said to you, yes. Just like taking blossom. Create it, you can create it differently.
No, it does not change. It's okay. It's a thing.
You understand?
See.
Just like any one.
For you were thinking that you don't know.
Yes.
You with some diseases.
Let me see.
Now you take a chance.
There's one
You
thing
incubate
that's
it. So you incubate it just like you have in plants. Growing. The time they do is like in culture and
There's nothing. What is
for animal.
That's not the way you use it.
That's how
Or
it
if
is.
you want to
Yes,
identify any diseases from animal,
Okay.
that is for those that working in the lab or in the mouth.
Yes, eventually.
Those that is working in the lab for the animals, you know, they take blossom. Yes, yes.
From the vendor.
Yeah, I've not.
For those this one, those want to be
Okay? So the dry screen method, are we listening? Yes. Dry screen is going to be for 6 minutes at 18,000.
Can we have something else?
I don't think you have a simple machine that's not subsidized.
And you
You
continue to
continue,
research.
yeah, actually.
You
Or
continue
you
to
continue
research.
your extraction. The thing is, it's just to optimize. It's not like it's too much. There's one person I'm waiting for. So if you don't place the caps properly, it will close, but it will break. While it's spinning, because of the speed,
You can't
it will
crash.
break. 6 minutes. For 18,000.
Usually we have lights. When there's lights, no, it's
That's
because
why they government to see. You find the meaning.
There has to be a way.
Around 24 hours lights.
you see some
Generator.
they will not burn the generator for the lab. And you want to do kind of testing. How do you want to do that?
It is responsibility of the researcher to find the meaning of the Like my boss now, my boss has to like
Yes, let's go back to the business of the day. We're not talking about Nigerian power now. We're talking about DNA. So far, is there anything you've observed from start or from start to where we? Is there something different we did not discuss? Dolako, do you have any question? Mr. Anyakola, yes. Madam Kumi? Okay.
I call this the dry skin.
Yes.
Something that for dry skin, there's no fluid.
Yeah, there's no fluid, that's why it's dry. There's no liquid. Any other question? Madam Edith? I know you're again, like just writing. Do you have any questions?
I like the dry.
Okay.
Yeah.
So we'll wait for it to
It's not
stay.
in the protocol.
It's not here.
Or you're just
trying to.
So like I said, once you're very familiar with your extraction procedure, you can actually optimize to give you the best yield you can. Okay. And of course, if you're not very good with optimizing, you best just stick to what the protocol says. Definitely.
I remember when I started working with the fungi bacteria,
Yes.
after we had done that, I find we transfer.
What kind of cell structure do you use for bashing?
It's actually this side that we actually repeat. This white
So what instrument do you use for the bashing? You know there's the lysis step that you use the bashing with. Okay, so with the check, let me look for the protocol so I explain what you're doing.
Yes, definitely.
They're using gloves for your phone. Against our rules and regs. Okay, so if you check this first stage, it says add 50 to 100 milligram with the fungal or bacterial cells that have been suspended up to 200 micrograms per minute. It says secure in a big beaker fitted with a 2 mil tube holder assembly and process at maximum speed. What big beaker do you use? So it's a different kit. It's a different kit. I'm just trying to.
After adding the only thing, we use the centrifuge.
You don't use a big beaker. That's right. Okay. So your license, there's no proper license. Yes, so there are big beaters. There's the if you check here. So say for example, processing time may be as little as 3 minutes when you are using high-speed cell disruptors like the portable sterilizers, sample processor, fast spread, 24, or similar. Or as long as 20 minutes when you're using lower speed, like disruptor Genie. We have a disruptor Genie, but it's not close enough for Yes. So it has a 2 mil holder assembly. So using the disruptor Genie, we use as high as 20 minutes. If you're using even vortex, can serve as. So if I'm even using the vortex, I'll go as high as 25 minutes. To just ensure that 20 minutes. To just ensure that lysis, yes, you know, the more the kinetic energy, the more lysis occurs. So using these small ones, you will have to use high speed to ensure that lysis is yes. So there, I'll show you what the fast step is like when we go. It's a big instrument, and the agitation is with vibrating energy. I use it during the muscles. Yes.
I think I worked with what I was in Africa.
It's something else. But in less than 5 minutes, everything is done.
Okay, so you're saying that 20 minutes you're not really doing your
Using a vortex.
Yes.
Yes.
Even the 20 minutes, you still have to be crying. When I'm holding it.
Yes, their hands. Exactly. But if you have a 2 mil holder or something like that, you just put it. Because this is like a vortex. The only difference it has a 2 mil holder assembly. Yes. Any other questions? Alright, this is almost done. And we will now add our elevation buffer DNA. Remember I said after we do DNA extraction, we have to measure the what? The purity and the concentration or quantity. Okay, that's what we're going to do after this. When it is considered pure, when it is pure, what's the 260-280 ratio?
1.8 or 2.1.
1.8 to 2.1, yes.
Or 2.0?
2.0, yes. When it is not impure,
280, 230.
It's going to be less than, it's going to be less than
230, 280, 230 ratio is what?
2 to 2 and
2 to
2.2.
2.2. Yes.
You know, yesterday you were not so particular.
No,
You didn't mention that.
I didn't mention that.
but it's inside the manual. Already
So we're adding to 50.
Remember I said nucleic acids have strong absorbance sites. 3 different wavelengths. Yes, exactly. At 3 different wavelengths. That's 260, 280, and 230 nanometers. So usually there's a ratio between 260, 280, and 260 and 230. So it's the ratio that you use to measure quality.
Okay, so the question I want to ask, we are saying
Okay.
whether we had water or
And it should go for.
solution
Yeah.
water. Or if we had water, there won't be any I want to put it, how do you want to put it? Because now, you know, the buffer is different with water.
The buffer can also be contaminated. So the impurities can also be introduced.
Maybe the question he wants to ask is why preference of buffer
About water, that is kind of highly
They've adopted it
It's
for
from
kind
the
of
key.
sample.
Okay, so
Yes.
it's
So if you don't have enough, so let's add the ratio of ratio
50
microliters. Check. We have the 20 to 200 microliter, 20 to 200, check.
No, I'm looking for one that I don't know. What is 200?
In 20 minutes, like our
so you're looking for something. Are we using the picture?
Sure, certainly. Usually what we do is we send a link containing all these facts, the training slides and the pictures. Just
I don't think that we have to change
Okay, the one in So is there somebody who
yeah, sure, there's so many examples. It's actually not a hands-on session.
Into the same column and
Yes.
Who was added? Have you added? Yes. Alicia Buffer, yes? Yeah.
I think we're supposed to time it, right?
Time what?
And
Yes, so where's the timer?
is everybody done? No, I don't scientific my everything. We're just leading a full temperature for 5 minutes.
Just here, that's on the bench.
I think maybe the right.
Yeah, we need my
It's just 1 minute already. Let's assume that we've done 1 minute.
The protocol here says we should
Yes.
Because
What percentage? Different kids with different protocols. Any other questions? So the final step is how many minutes? At what speed?
Maximum.
It should be maximum 1 minute, but the maximum speed is 1 minute. The rotational is not included.
Yes, the rotation.
It's a maximum speed for 1 minute.
Let's do
That would be
18,000.
18,000, yes.
If you have questions, speak.
I'm just trying to imagine if you don't have the microcentrifuge that has the required.
It's been difficult to achieve. The thing with centrifugation now is to help you get rid of as much weight impurities as possible. Remember. So if you don't centrifuge, it is very likely that your DNA will be impure. Please check if there's anybody. Maybe they're not on. So the essence is to get rid of impurities. If you don't have a high-speed centrifuge, a good centrifuge, it means that you may not be able to get rid of all the impurities, and that simply means that your DNA will be impure. Okay, it's just something when you come back. Or rest, please. So usually any instrument that is not in use should always come from the source to avoid any issues. Thank you. As long as it is not in use, it should remain. So we have how many minutes? Well, okay. Okay, you were not here yesterday.
Positive displacement.
It's a positive displacement. So it doesn't use the normal tips. If the tips it uses are the ones that look like in syringes. So you keep it here. So what you do is set it at the volume you want. So if for instance I set it at 10 and maybe I want to dispense 10 times. Do you understand? I'll set it and I'll keep into whatever I'm. When I'm done, if you notice, it keeps going down. So once you are done, and it will also be counting, it will display the number of times you've dispensed. So when you keep, notice. Once you are done dispensing, get back to this position and you eject the tip.
So it has a maximum.
So this is 1 microliter to 10 ml. You can't do beyond 10 ml. This you take it back home. So for every time you dispense, this goes down. Goes down again, goes down, goes down. So if you are trying to dispense a particular volume several times, it's nice to use this. Yes. Oh yeah. Let's have your DNA. Properly capped, please. We will now have a moment of truth.
What that we don't want us to say clearly.
The truth of
There's
the
nothing
DNA.
leading. There's nothing leading now.
Next.
That's more.
So is good. It is a blessing. Praise 1 minute. Hello. Hallelujah.
Then I'll move to this year.
We'll move to this year first, before PCR. Then after the PCR hands-on session, we go downstairs for the geroelectrophoresis theoretical session. First thing tomorrow morning, or evening, we'll have sequence. See how things proceed. That's what we call it. I expect that we are here before 9 tomorrow. That is okay. Let's be here before 9. So that we don't have to spend too much time. Like now, we came in some of us came in. I understand that not all of us were aware of the time. The second, we still need to time of something.
So what's the time?
9 o'clock. 9. If you can come in before 9, say 8:50. Or we now proceed. Let me just take a so someone can remove it. I'd just like you to. So take out your spin column. Take out the spin column.
I always switch
See. Just color. Alright, discard all your used tubes. Discard everything. The ones that have liquid, discard into the appropriate liquid discarding.
You can probably check once. Already discarded.
So who doesn't have?
Can you discard everything?
I said your spin column alone. Your spin column alone. Discard your spin column, yes.
And the one
What
that
you
has-
have in your tube is your DNA. Your spin column alone. Your spin column alone, please.
All these ones, I can't remember. I did not told me. I can't remember. Now this one is looking used.
Yes, please discard. Or you have your DNA. Okay, please discard every other thing.
Yes.
But you call the water.
What one did you discard?
Oh, we didn't pour water available. Is the column you're training?
Okay, you also discarded this too. This is what I want to play now. No, not this. You've already removed again that already bonded to this silica gel.
Something is like
Yes, now that is where we have the DNA.
But this one can work.
Okay, you give
You know it's plenty
More than this. How? Is that not the
No, it's supposed to be small like this.
I just hold it.
You treat the work?
No.
All right, so you're going to check for purity, the purity of the quality of your DNA, right? Using your spectrophotometer, that's the nanodrop. Like I said, this is a nanodrop, this is a microvolume nanodrop. There are two types, there's microvolume and there's a cuvex type. This is microvolume, so you can use as low as 1 microliter of some. This is the arm. This is the arm. Okay? This is the arm. So when you want to take a measurement, dispense your sample onto this surface, very tiny portions aperture. Lower your arm gently and measure. But before then, you must always use your elution buffer to blank. So remember, our option is double-stranded DNA plus dsDNA. You click it. Okay? Do not click some instrument is doing self-test. When it is done with the self-test, we will volume. Please, we need pictures. Let's clear our next. Anything that's continuous, we will need this. We will need a very small pipe. Let me see the other one. These tips. So look at this now. You can now blank. Remember, okay, I didn't mention that. So what we use is link-free webs. Link-free webs. Link-free webs and link-free papers or something are the ones that don't leave particles. You know, the common tissue, they leave this small particles, which is not advisable for this. So they are called Kim wipes. Kim wipes. So we use them to clean. That's the ideal thing to use. But just ensure that whatever you're using to clean is link-free. Particular time we supply Kim wipes. But ensure that it is link-free. So that's the idea. Lift your arm. Clean your pedestal. So it's clean both pedestals. So these very tiny portions are called pedestals. My lights is not in my eyes. Pedestal. Yes. So we take this. Let me have the elution buffer. If you can't use your one hand, because I'm not eating my hand issue.
Why are we using this?
Yes, to blank. So now let me also explain something. So this, look at it, it is set at 2. Remember that the same way you do your gel, you will not press to your second stop. You will always press to your second stop when you are dispensing. In this case, you will not. Because if you press to your second stop, you will take up what you have on the surface. So what you do is press the first stop. Once it gets to this place, remove your hand and discard your tips. I'll take it again. So when you are dispensing, especially when you're working with the nanodrop, do not press your micropipette to second stop. It's best to press to first stop, okay, and take it out. If it's possible for us to just use this because of the so do not press to second stop. Press to your first stop, okay, and remove your hand, discard your tip, and lower your arm and measure. So you must always clean your pedestal, which I've also already done, and lower your arm gently and blank.
Okay, so what quantity did you actually use? What quantity?
So in the actual sense, this is 2, but because we don't press to second stop, not the whole volume leaves. So let's say 1.5 to 1 to 1.5 microliters is what gets to the pedestal eventually. It says clean both pedestals and load. The blank is okay. The blank okay, and you can proceed. So you clean the pedal. I'll just try one, and everyone will come and do this. We have one side. Measure. So let's see the quality and what concentration of the Oh, this is good. So take a look at this. The concentration is 282 nanograms per microgram, which is very sweet. The purity is 1.91. Remember we said 1.8 to 2.1 or 2.2. This is perfect. The second one too, which is something I said we can always neglect, is also good. So this is ideal. So we will change whose own is this. So Bolly, what's the name of the sample you want to use? Volume.
Yeah, from one.
Okay. Thank you.
My was me already.
Okay. So I'm ready. You're ready. They're ready to take. So you can just be taking them as they. Oh, don't worry. I'll do it. So everybody, please ensure that you don't block. So you load your sample gently and lower the arm. Always clean and clean. So the next person can load.
No, we can't. There's no need to blank now.
Because you use the same elevation buffer all through. So let's quickly do that. And
You
proceed.
cleaned already?
Yes, I cleaned.
You have to have so maybe just put this space for the video.
So do me a favor. That phone will affect what I'm going to do now. So just.
My bully is not saved. Oh,
Still on the screen.
still on the screen.
Remove it and put your name.
It's already saved like that.
Press.
It has not
It does not.
Have you measured?
No, there's no measurement on the screen. You just wrote Bolu and there's no icon for
no,
it.
so you can say those.
Go to that
No tweezers.
Hold on, come.
I still have my result on this.
Have you
Yes,
labeled?
no, she hasn't been here.
So should I?
Load two.
Yeah, I clicked on it.
Load yours. Have you loaded?
She already
Measure.
loaded.
Where do I click? Where do I press measure?
Maybe.
Not bad. Concentration is low, but purity is not bad too. But even for 39.6 is not bad. So label, change the label to your name. Sorry, adjust the color of your laptop, Madam Eddie.
Have you said need to what?
Open it.
Lift the pedestal. Gently. Gently to mount. Do the same for the upper pedestal. Yeah, so let the next person step up. Sorry, please move back a bit. Your phone, please.
You have, take it.
You have.
You have, please take it.
You have now.
Your phone, please.
Yes.
Lower the pedestal.
And you press music. Interested in saving in 1.3
Pretty low. Slow. Next.
So what is it, sir?
You can give me sample.
You are done. Are you done like that?
No.
Okay, just press down.
Done.
And the next person. Who's the next person?
No, I don't need it in last person. Remember to do this problem.
Something.
It's for you.
Where is it?
So sometimes my feet is sample.
How do you use the
Good one.
old one?
Sorry, sir.
You can get it in any supermarket, okay?
Yeah, I don't see key wipes. You see wipes.
Lint-free wipes.
It has to be lint-free.
Linked.
Lint.
Lint are
L-I-N-C.
those tiny particles, like if your clothes get old, very tiny. Doesn't lint anywhere, it's very sugar. First,
Someone help her take the tube. My own phone, no pocket.
careful. Careful. Yeah.
Measure. Quantitation is low, security is low.
Okay, what's the one with person? Six. So it's only one person that has had very good parenting. Maybe because he has been doing it.
It depends on the organism.
Depends on
Depends on the organism, that's true. And I also mentioned that when you are taking from your plate, take as much as
It took a
It took,
long time.
yeah.
Even when we were testing, it was not easy.
Yeah,
It
it's not
was-
too
It
careful.
was
That is a but.
at the bottom. It was not easy.
You
for
can't
them to
force
use.
it.
End of the
What did you do?
what's that?
You said end of the experiment.
You break on that.
No bother, it's fine. We're not going to leave it. Go back to the experiment. Alright.
But did she say?
Did you type the
Did you
name
add
of
the
the
organisms?
name of the organism?
Just my name.
She just
Please add it,
found
add it, add it.
What is it?
And it's
Like that.
Done. Next person.
remove your
Sorry, sorry.
appliances. All the way from her. Not easy.
So then when must now who will take care of it? This is not the use your own thing.
Hey, please don't leave me, tag me. I'll send you my
So when you're posting anything regarding your workshop, ensure that it's an AfricaBP.
I did that last week.
I did that too.
I think it was same before, isn't it? Mine came before yours.
Tag of us, of course, but ensure that it's an African Genome Project.
VP.
Yes. Yeah, I think they're handled on in LinkedIn. It's African Biogenome Project. Yeah.
I'll just do them how we do them like this. Thank you.
Well,
We're running behind schedule, project, so let's be fast. It's almost 3:30.
Seriously.
pressure. Are you major? I always high a little bit. But you still look.
That's good. The concentration is good. Security is good. Yeah, it's good. So let's end the experiment.
So now we take it. It was just snapping on. I'm not snapping on.
Yeah, I'm not snapping. I'm just doing.
They're more
Please, don't worry. Okay, Paul, please, I want to do PCR now. Now, please.
Please, the camera is at your back now. The camera so that we not fall down. Yeah, down.
It's just a second one. What are you guys doing? Can you go home with it?
Yeah, no, I'm yeah, you have done that one is not true. That's the first one.
Shall we proceed? Shall we proceed, please? Next.
He's just taking pictures of Jane. He wants to get home.
Here's your phone now.
It's okay. You don't know already.
Phone now, just slowly.
Can we end the experiment? Then switch it off.
And we will save it in the flash first.
Why don't you measure it?
No, mine was the first.
That's the first
He
one.
was doing a demo for the picture.
Just for the
That's what I was
picture.
telling you.
No, she's the one that
This is the concentration of the DNA. It's high.
I think from 20.
the guys are pure.
And the protein is between 1.8 or 1.2 years. So she has the highest spirit.
What is the
No,
Yes.
so let me see the way we work.
I have the highest
I have a
spirit.
friend for a colleague that did a research. Her name is Inqaba Biotec. She's from Reno. So still working with it.
So whatever.
We did raise the volume more.
Just whatever you need to say. Okay, so I think we're done from here. Just tidy up. Bolu. Sorry, not you. Bolu girl.
Holy boy.
Please help me with
You want to run it?
no, the flash.
Okay.
Okay, so let's get started.
Have you picked it?
Take one.
Okay.
Ma, are we still carrying it?
No, for the PCR. I don't want to go
Why is it the idea of
PCR.
Camera, I don't know.
So you need to PCR.
So you want to do it now? Yeah, should we probably do by night? Can we run it more now? Or you just wait and leave it one night? Maybe focus and help practitioner.
Alright, let's start the Okay, so right now we're moving to the main lab to have PCR session. Okay? So let's relocate.
Same that workshop, right?
AfricaBP workshop.
African.
BP workshop.
But we need
So
virtual
where what of the rest of the samples?
How many pictures last night? No.
Okay, so let's move. Okay, guys.
You're packing it. Please, these samples are for the PCR. I think we're supposed to put them together.
Yeah, you're supposed to be sure. No, this is not what I'm talking about.
So that this was
Ingredients I'm talking about.
So where are you going to put them? What of the others?
So, yeah, heading straight to PCR now, right?
Are they